For today's practical session, we'll be looking at some of the biochemical tests that we use for bacterial identification, how to carry out the test, and the basic principles of the test. So for the first test is the Indole Test. So Indole Test is part of the IMVic test. IMVic test stands for Indole Test, IMVic. So I stands for indole, M stands for Methyl Red, V stands for Voges-Proskauer, then C stands for the Citrate Utilization Test. So it's like IMVic stands for represents four tests, four different kinds of tests that is very useful in the identification of gram-negative bacteria. So for indole, we use tryptophan broth, okay, a tryptophan broth, and we inoculate this tryptophan broth with the bacteria. So we are looking for bacteria that can break down tryptophan into indole. Okay, so the bacteria essentially has the tryptophanase enzyme. Okay, so after 1 day of incubation, we will add 4 to 5 drops of the coax reagent. Okay, which will will shake gently, and after a while you can see a red ring formed on top of the solution if the test is positive. Okay, so this is a negative test, this is a positive test. So this is useful to separate Escherichia coli, which is indole positive, versus other bacteria, and for example Enterobacter, which is indole negative. Next is the Methyl Red and Voges-Proskauer test. So for this set of tests, we'll use the same broth, which is the Mrvp broth. Okay, so we are essentially looking at how the different kinds of fermentation and end products as the organism utilizes glucose. So for Methyl Red test, after 2 days, we'll add 5 drops of the Methyl Red reagent, and if it is reddish, it is positive. So what we are looking for is the mixed acid fermentation, where the bacteria ferments the glucose and produces stable acidic end products. Whereas for the Voges-Proskauer test, we are looking for bacteria that ferments glucose, but then at the pyruvate stage, it will produce acetone as an intermediate, which will be further oxidized into diacetyl and butanediol. So when we add the reagent, which is the alpha naphthol reagent, into the Voges-Proskauer tube, it will react with the acetone and then form a reddish color. So this is positive for VP, and this is positive for MR. So we usually do this together. So Mrvp test to see whether they produce a stable acid end product for MR positive, or a or it produces 2,3-butanediol fermentation, which is for VP positive organisms. So because the change in pH sometimes requires time, so for this kind of test, we usually test it once after 1 day, where we remove a bit of aliquot from the test tube and then place it in a clean test tube and then add the reagents to see whether it is already whether the acetone has already been produced. If it is negative, we will test again after 1 more day of incubation. So the testing is done after 1 day and after 2 days of incubation. That is for Mrvp test. For citrate tests, we usually utilize Citrate Agar. A popular one will be the Siemens Citrate Agar. As you can see here, the only nitrogen source is the ammonium dihydrogen phosphate, and the only carbon source is sodium citrate. So what happens here is the bacteria has the enzyme citrase utilizing citrate, okay, and produces oxaloacetate and acetate products. So as it utilizes the carbon here, it will also metabolize the ammonium salts. So this will release ammonia and also sodium carbonate, and this actually increases the pH of the medium, and it changes from green to blue. So blue is positive for Siemens citrate agar, whereas green or no change is negative. Next, we look at oxidase test. So oxidase test is where we look and to see if the bacteria has the cytochrome C oxidase. Okay. The reagent is tetramethyl P phenylene diamine. This is a colorless reagent. So usually what we do is we will place a bit of the culture onto a filter paper and then drop the reagent on top. Okay. And another way is to moisten the filter paper first before smearing the paper with the bacterial culture. If you see a deep blue or purple color, that means the bacteria has the cytochrome C oxidase and is able to oxidize the reagent. Okay. So the point to note here is you cannot use your normal wire loops to pick your isolates. You have to use a wooden applicator, usually a sterile toothpick, or a platinum loop. And because the platinum loop is more expensive, we will usually stick with sterile toothpicks. Okay. We cannot use the normal nichrome inoculating loop because that will cause a false positive as it will react with your reagent. Okay, so this is an important test as it differentiates between the oxidase positive bacteria, for example Pseudomonas aeruginosa, your Vibrios, and from your E. coli, which is negative. Another important test is the Oxidative-Fermentative Sugar Test. Okay, so the OF glucose test is where you use usually we use a phenol red broth and we add glucose into it as its sole carbon source. Okay, once we have inoculated a pair of tubes, we will overlay one with sterile paraffin liquid. Okay, so this will ensure that the oxygen tension here is low. Okay, so from after 1 day of incubation, if you see only this tube is positive, which means the glucose is fermented in this tube, is utilized in this tube with acid products, but there is no reaction here, that means the bacteria is only oxidative, for example Pseudomonas. However, if we see both tubes having acidic pH, okay, that means this bacteria is a fermentative bacteria. It is able to utilize glucose both conditions, whether you have oxygen or when you don't have oxygen. Okay, so if you look at this diagram here, it shows the importance of both oxidative fermentative glucose test and oxidase test in order to differentiate the families in the class gamma proteobacteria. Okay, gamma proteobacteria is important because this is the class where most of the bacteria can be cultured. Okay, so this is the order. We have Enterobacteriales, Vibrinales, Aeromonadales, Pastorales, and Pseudomonadales. Okay, and from here you have your family. So if by just utilizing the oxidative fermentative test and your oxidase test, you will be able to separate up to three different families. So Enterobacteriaceae is oxidase negative. Okay, whereas for Vibrinaceae it is oxidase positive. Pastoralaceae is oxidase positive. Pseudomonadaceae is oxidase positive. However, between Vibrinaceae and Pseudomonadaceae, you can see that Pseudomonadaceae is oxidative, whereas Vibrinaceae is fermentative. Okay, so from here you can differentiate Vibrinaceae from Pseudomonadaceae. So between Pastoralaceae and Vibrinaceae, it is whether it is motile or non-motile. Okay. Next is the Catalase test. So Catalase test is where you place some culture onto a glass slide and then you add a drop of catalase reagent. The catalase reagent is usually 3% hydrogen peroxide. So if the bacteria has the enzyme catalase, it will degrade the hydrogen peroxide and form bubbles, which are the oxygen gas. Okay, so this is a positive reaction for catalase. So it is important in bacteria that grows in aerobic conditions because uh, utilization of oxygen actually produces hydrogen peroxide, which has to be reduced to less lethal forms of water and oxygen. Look at the coagulase test. So coagulase test checks whether the bacteria has the coagulase enzyme. So coagulase enzyme is important in pathogens because it works with the plasma components to form protective fibrin barriers around the cells, bacterial cells, and protects them from phagocytosis and other types of attack from the human immune system. So what we usually do is we will put the plasma, we will mix the plasma and the bacteria together. So there are two ways of doing this. We can actually do the slide test or do the coagulase tube test. So if there is coagulation, if there is a reaction, a positive reaction, so the bacteria will be clumped and you can see particles of clumping of bacteria here. So this is actually negative where the bacteria is actually looks like a smear. Okay, whereas if you use the coagulase tube test, if it coagulates, if the coagulase test is positive, you can see that it remains and not liquid as in the coagulase negative test. So coagulase test is usually very important to differentiate between coagulase positive staphylococcus versus coagulase negative staphylococcus. Next, we'll look at the hemolysis on blood agar. Pathogenic bacteria usually can also lyse blood. Okay, blood lysis can be categorized as beta hemolysis. So that is clear zone surrounding the bacterial colony, or alpha hemolysis, where there is lysis, slight lysis, but it is only slight greening and not as clear as this one. There's another category which is gamma hemolysis, which actually also means no hemolysis. Next, we'll look at the triple sugar ion agar. So this is a very important biochemical test now for the identification of bacteria, especially gram-negative Enterobacteriaceae. It is able to differentiate bacteria on the basis of glucose fermentation, lactose fermentation, sucrose fermentation, and sulfur reduction. So all this in a single tube. Therefore, it is very, very useful. The media is performed, prepared by preparing a shallow agar slant with a deep butt. So what we mean here is the slant is shorter, whereas the butt is longer. We do this so that we can get some anaerobiosis at the bottom of this tube. So what we see is the fermentation of the sugars, the differential fermentation of sugars between among the different bacteria. Okay, if the triple sugar ion agar is inoculated with only glucose, only fermenter. If TSI is inoculated with a glucose only fermenter, the acid products that are produced will lower the pH, okay, and turn the medium yellow. However, it is only if it is only a glucose only fermenter, the glucose will be quickly utilized and then the bacteria will start utilizing other products, for example, the amino acids. This will instead start producing ammonium and raising the pH, especially at the slant area. So this is called a reversion, but it is only limited to the slant because of the anaerobic uh, conditions here. Okay, so the breakdown of ammonium products, ammonification, requires an anaerobic condition. Okay, an aerobic condition. So glucose only fermenter will give you this kind of results where there is a yellow but a alkaline slant. Okay. However, if the bacteria can ferment both glucose and lactose or glucose and sucrose, then the whole tube will be yellow. You can see there's a break here also. That means the bacteria is producing gas, breaking up the agar here. And for this, it is a clear-cut H2S producer, hydrogen sulfide producer, forming a black region here. You can see a slight reddish here, so that means it is also a glucose-only fermenter. So probably this is a lactose-negative, glucose-only fermenter producing H2S, and therefore it is most likely a salmonella. So the differential reaction we see here is because of the different concentrations of sugar. Dextrose, which also stands for glucose, is only 1 gram per liter, whereas for lactose and sucrose it is 10 gram per liter each. Okay. So this and the combination between ferrous sulfate and sodium thiosulfate, this will enable the media to detect for hydrogen sulfide production. Okay. So this is another media that is actually a precursor to this triple sugar iron agar, the Clickers iron agar. Here there is only 2 sugar, lactose and dextrose. The ratio is also the same, dextrose in very small amounts, only 1 gram, whereas lactose is 10 grams. So for here, you know for certain it is a lactose fermenter. This is only a glucose fermenter. And this is a glucose only fermenter and also H2S producer. You use a TSI tube, you using a step instead of a loop, using a step, you streak first and then you step the TSI slant. That will ensure inoculation at both the butt and the slant areas. So these are the different results that you can get and the interpretation of the results. This is the symbol that is usually used to represent the results obtained from the TSI agar. Next, we'll talk about the potassium hydroxide string test. So this is a string test. What you do is you emulsify some of your bacteria with 3% potassium hydroxide, and then using a loop you see whether the cell emulsion has become viscous and stringy. So if it's viscous and you can see a positive string test here, that means the bacteria here is a gram-negative bacteria, as gram-positive bacteria will give negative results. So the utilization of string test has also been expanded by using a 3% sodium deoxycholate solution on a glass slide. Okay, the observation is still the same. However, this is very useful for the identification of Vibrio cholera. Okay, Vibrio cholera as opposed relative to the other Vibrios. So Vibrio cholera is positive for string test if you're using a sodium deoxycholate solution. Next, we have the amino acid decarboxylase dehydrolase test. Okay, usually for this test, we'll run it with molars decarboxylase broth. We'll inoculate the bottles with 1% arginine, 1% lysine, and 1% ornithine, and also one control without any amino acid. Once we have inoculated the bacteria into the bottles, we will overlay the broth, all the bottles, all the four bottles, and then we'll incubate for up to 4 days. And we'll look for positive results. So the positive results will be purple to violet, whereas the negative results is yellow. So what happens is, after inoculation, we'll utilize the glucose, will ferment the glucose in the anaerobic medium and turn it yellow. Okay. And the utilization of glucose will then stimulate the decarboxylase positive organisms to start producing the enzyme. So the decarboxylation of amino acids will start the accumulation of alkaline end products. For example, the amines. So because this is an alkaline product, the pH indicators will start turning to purple. So purple is considered positive. Okay, so purple is considered positive. This is the uninoculated control, and this is the negative. So it is negative is yellow because the bacteria utilizes only the glucose and reduces the pH. Okay, but if the bacteria also has the decarboxylase enzymes, it will further break down the amino acids and produce amines that will increase the pH. That is why when you inoculate the decarboxylase, amino acid decarboxylase and dehydrolase test, you must always do it with a basal medium pair. That is why you have to always carry this test out with the basal medium without any amino acid. Okay, because if the organism is a glucose fermenter but does not produce any decarboxylase, the medium will only turn yellow. But if not, it will turn if it has decarboxylase, it will further turn purple. If you do not have this control, you will not really know whether your bacteria is growing well or not. Okay, therefore it is better to have this than to have this pair of results where your basal medium will be yellow and the one with the amino acid will turn purple.